Now, howdy, y'all. We're going to be talking about the Central Dogma, Part 1, which is the history of DNA. So, we all know now that DNA is the genetic material, but we didn't always know that. So let's look at the history. So, first we have bacterial transformation. It was done by this guy named Frederick Griffith. He used Diplococcus pneumonia to infect mice. Now, Diplococcus pneumonia is going to be a bacteria. And he used that bacteria to infect mice, and it caused the mice to get pneumonia. Now, there were two types of bacteria that he used from Diplococcus pneumonia. There was a rough bacteria, or an R bacteria, that had a rough coat on the outside, and it did not cause pneumonia. But he also had a smooth type, or an S type, that had a capsule around that bacteria, and that one caused pneumonia. So, the coat was associated with the virulence, and virulence is the ability to cause disease. So let's take a closer look. When we injected the rough, non-virulent into the mouse, it lived. But that smooth, virulent strain that had a capsule around it, when we injected it into that poor little mouse, the mouse died. Because it caused pneumonia, and that mouse died. Now then we took that smooth, virulent strain, and we heat killed it. Now when you heat kill something, you bring it up to really high temperatures, and it typically bursts because it's so hot. So, when we injected that burst, smooth, virulent, was dead into that mouse, the mouse lived. Now this is where things get kind of funny. We took live rough and mixed it with smooth dead. Mixed them together. We took that concoction and put it into a mouse. Well, if the rough allowed the mouse to live, and the dead smooth allowed the mice to live, we would think that we would allow the mice to live. That's not what happened. When we combined the two, the mouse died. And when we looked inside the mouse, we actually found live, smooth bacteria. Well, how did that occur? Well, the living rough type was able to take up the smooth type genetic material, and the rough type used that genetic material to create its own capsule. And once it had created a capsule, it had become a smooth type of Diplococcus pneumonia, and therefore it could cause pneumonia. So that's where we came up with the idea of the transformation principle, the ability to take up and use foreign DNA. But again, at that time, we didn't know that it was DNA. So, Avery, McLeod, and McCartney took this experiment one step further. You know that concoction that we had? We had the R, and then we had that heat-killed S that kind of exploded everywhere. Well, in that concoction, they split it into two vials, because they wanted to see whether the R bacteria would pick up protein, or would it pick up DNA? So, in one vial, they added protease. Now, protease is an enzyme, because it ends in A, and it degrades protein. So all the proteins that were out there were degraded. And in the other vial, they used DNase, another enzyme that would degrade DNA. Then they injected both of these into mice and saw which one died. Now, the protease would just eliminate the protein. The genetic material was still there. So the R bacteria took up that genetic material and used it to create that smooth coating, and we saw that the mouse died. But the DNase took that DNA and degraded it, and then that R bacteria could not find any DNA to take up and use, and so it could not become an S bacteria. So this mouse lived. So they determined that it was DNA that was being taken up and used for our transformation principle. Now, we do know that DNA is the genetic material, but many people didn't want to believe the scientists at the point in time. So, then we had Hershey and Chase come into the picture. They again were trying to prove that DNA is the genetic material. So they took a virus and they infected injected E. coli with that virus. What they did, though, is they went ahead and knew that that virus had a protein head and a DNA core. But they weren't sure what the virus was injecting into our E. coli or our bacteria. Now, they knew that it was injecting the genetic material, but they didn't know what the genetic material was. So they did two things. They took a batch and radioactively labeled the sulfur on the protein, and they radioactively labeled the phosphorus in another batch, which is in the DNA. They mixed those viruses with those bacteria, let them inject the genetic material, and then they looked at the bacteria. And in one, they didn't see any radioactivity, and in the other they did. So let's take a look. So, when they radioactively labeled the sulfur, it just labeled the outside of our virus. So when the virus came in and injected the genetic material into the bacteria, it didn't inject the capsule on the outside. It injected the DNA, and the DNA was not labeled. But over in this batch, where we radioactively labeled the phosphorus, which is on the DNA, when they looked at the bacteria, they had seen that the virus had injected the DNA. So again, DNA is the genetic material. Now looking at the structure, we knew in 1909 that DNA existed, that it had a sugar and a ribose backbone, and that we called it deoxyribose sugar, and it has 3 parts to every nucleotide. A nucleotide are going to be the polymers, or excuse me, the nucleotides are going to be the monomers that make up the DNA. We have a sugar, a phosphate, and a base. We see our subunits are composed of the sugar, which is a deoxyribose sugar, a phosphate group, and a nitrogen base. Our nitrogen base is one of 4 different types. We have the purines, which are adenine and guanine, which are the larger bases, and we have the pyrimidines, which are thymine and cytosine, and those are the smaller bases. You can know this and remember this because the name of the type of bases is opposite to the size. So again, our purines with the small names are going to be the larger bases, and our pyrimidines, which are the long names, is going to be our smaller bases. And again, we have a phosphate, a sugar, and a nitrogen base. Now, base pairing. Base pairing was figured out by this guy named Chargaff. Now, Chargaff figured out that adenines bond with thymine. So straight letter with straight letter, A to T. And guanine binds with cytosine, curvy letter with curvy letter. Now, how are they actually combined together? They are held together by hydrogen bonds. Adenine and thymine have 2 hydrogen bonds. Cytosine and guanine have 3 hydrogen bonds. So it's hard to break up cytosine and guanine. Now we all know about Rosalind Franklin, and she gave us a photo. It was an X-ray diffraction photo of what DNA looked like. She didn't get much credit at the time, but she has been given credit now. Watson and Crick used that photo and actually was able to come up with the idea of how DNA was structured. It is a double helix, and the 2 chains that create that double helix are anti-parallel, which means they run in opposite directions. Again, they run in opposite directions. One goes in one way, and one goes the other. That's why it allows it to twist, and we'll see some complications from this when we talk about replication, and that's what we have.